All right, hello everyone. Sorry I can't be there, but I did want to review with you the key for and the answers for the cell transport concept practice on the last page of the blue packet that I gave you on, what day is it, Monday? So the first couple are pretty straightforward. How is osmosis different from the diffusion? And down here I have the key. So osmosis is the movement of water across a membrane. It has to be the movement of water, and it has to go across the membrane, and it is always going with the concentration gradient. Whereas diffusion is the movement of any substance, also along the concentration gradient. Okay. So number 2, let's take a look at this. So if the concentration of solute molecules outside the cell, so it's in the solution, is lower is hypo for lower, hypotonic, and the cytosol is the liquid portion in the cytoplasm. That's not looking at the other dissolved stuff. It's just the liquid part of the cytoplasm. Let's see. Is the external, okay, so that one is hypotonic. And then here we have a picture. Is this working? It's working. So the picture here, we have the triangles. If it's lower on the outside, then I just drew 2, and I drew a lot of triangles on the inside. And so the water will move into the cell, making it bigger because the solute concentration is lower around the outside. All right. Let's see if I can move this one down. Okay, so number 3 is about the Dead Sea. So explain why you think nothing really can live in there in terms of osmosis. Appreciate you bearing with me here. So the Dead Sea is a hypertonic solution which causes the cells to lose water or to dehydrate. How you know it's hypertonic is I gave you the value 28 and 35% salt in the Dead Sea, whereas the human body, average organisms, not the human body, average organisms contain a lot more water, which means it's less dissolved solute. Okay, so this one is 25 to 10% dissolved salts, protein, sugars, everything else that's in there. So that's less in the organisms, more in the sea. So that's why it is a hypertonic solution and the water will rush out of those cells. All right, and then let's look at number 4. Number 4, you're going to see something like this on the test. I think it's a little bit easier than this one though. So let me look here. You're probably cringing right now, but that's okay. I don't know where it is. Okay, that works. So here we have a YouTube, and the membrane is permeable to water. So here I have my key. So water can go back and forth. Substance A can go back and forth, but B cannot. Wherever B starts out, that's where it's going to end up because either it's too big or it just doesn't get along with the membrane. So 40 grams of substance A and 20 grams of substance B have been added to the water on side 1. And let's see. Oh, here we go. So 40 grams of substance A and 20 grams of substance B have been added to side 1, okay, of the YouTube. And then 20 grams of substance A and 40 grams of substance B have been added to the side 2. So when the system reaches equilibrium, how many grams of substance A will be on the side 1 and how many grams of substance A will be on side 2? So they will each have 30 grams of substance A. Because substance A can go through the membrane, it will do that. So substance A will even out. Okay? Okay, that's that. Let's move it on down. So side 2. That's same for side 2. Okay, so when the system reaches equilibrium, how many grams of substance B, I'm going to move it up actually. How many grams of substance B will be in solution on side 1 of the U tube? How many grams of substance B will be in the solution of side 2? And explain. What will happen to the water level? And explain. So let's take a look here. I'm going to move this up so we can see everything together. Okay, so after substance A has evened out, side 1 will have 20 grams of B because it can't move, so it's stuck there. Side 2 will have 40 grams of substance B because it's stuck on that side. However, the membrane, okay, so this is why, that's the explanation. The membrane is not permeable to substance B, so it can't pass through. Whatever was there first will remain there because it doesn't pass the membrane. I hope you're seeing my little movement of my mouse. Okay, and then the next one, the water level will increase on side 2 to equalize the concentration of substance B. So because substance B cannot move, but the water can, the water will dilute substance B on side 2, and so it will increase. This is like the primo example of osmosis in a U tube. You will see this on your test. You will see this in college. So you're welcome. Okay, if you have any questions, you might just have to wait until I see you next, which is on Friday. So hold your questions, or you can reach out on Google Classroom, and I think my phone will ding me because I'll be out of town. All right, good luck. Have a great day. Maybe I can stop this thing from recording me.